So this is the second lecture of a series of lectures provided to give students an introduction to the immune system. The first lecture focused on defining the roles and types of different cells and molecules that are involved in immune reactions and their importance in health and disease, and also an introduction to the innate immune system and the acute inflammatory response. This lecture focuses more on the adaptive immune system, T lymphocytes and B lymphocytes, and the mechanisms that these cells use to recognize antigen. So the learning objectives for this lecture are: 1, to describe the similarities and differences associated with B-cell receptor and T-cell receptor recognition of antigen; 2, to compare class 1 and MHC class 2 antigen presentation pathways and associated immune responses; and 3, to recognize the importance of Major Histocompatibility Complex, or MHC, polymorphisms in regulating immune responses in human disease. So to give a big picture view of the importance of antigen receptors, this is a cartoon illustrating the importance of antigen receptors in 2 major steps of the immune system function and development. So the first part of this slide illustrates that all lymphocytes arise from a common lymphocyte progenitor that comes from a hematopoietic cell in the bone marrow. And these cells give rise to a large repertoire of immune cells, each of which that express a different antigen receptor capable of recognizing different molecular determinants. So in this cartoon, you can see you have one cell and a huge army of millions and millions and millions of different antigen receptor specificities. And the generation of this antigen receptor repertoire occurs during lymphocyte development and occurs independently of any antigen stimulation. The next part of this is when these cells move into lymphoid tissue in lymph nodes or the spleen, as we'll talk more about in a few minutes. They will come in contact with antigen, and based on the antigen receptor specificity of these cells, it will specifically interact with a molecular determinant of that antigen, and that cell, in this case the yellow cell, will become activated and will proliferate. It will make more of itself and will also differentiate to develop immune effector function, and that's all specific for that one specific antigen recognized by this one yellow cell. So structurally, T and B lymphocyte antigen receptors belong to a family of molecules that are termed the immunoglobulin supergene family. And you can see here in this picture, you have a cartoon of the B-cell antigen receptor and the T-cell antigen receptor. And in both cases, they're made up of heterodimers, meaning they're made up of 2 different protein chains that are associated with one another by interchain disulfide bonds. And they also have a conserved intra-chain structure also defined by disulfide bonding that forms these little domains or loops that are referred to as immunoglobulin domains. So antigen receptors are key to the functioning of the adaptive immune system. They're very specific, and they're required to generate an adaptive immune response. As I mentioned earlier, T cells and B cells have structurally similar antigen receptors, but as you'll see in a few minutes, they recognize very different forms of antigen. And so just to define what we're talking about here when we're referring to antigen, it's really a circular definition. So antigen is really any molecule or part of a molecule that's recognized by the antigen receptors of lymphocytes. And an epitope is the piece of the antigen or the molecular feature of an antigen that's specifically recognized by a lymphocyte antigen receptor or antibody. And so this is a cartoon to kind of illustrate that concept. So in this cartoon, you have 2 different antibody molecules, which are really the secreted form of the B-cell antigen receptor. You have a blue antibody molecule up here and a red antibody molecule down here, and in the middle is an antigen. And what you can see is that the blue antibody molecule recognizes one portion of the antigen, and that portion of the antigen it's recognizing would be referred to as the epitope, while the red antibody molecule recognizes a different molecular portion of the antigen, and so it's recognizing a distinct epitope. So they can so different antibodies or different antigen receptors can recognize different molecular features on the same antigen. And so again, to give a little bit of an overview of the structural features of antigen receptors, in this case we're focusing on the immunoglobulin molecule, or the B-cell antigen receptor, you can divide up these molecules into two main regions. One is on the amino terminus up here that's very, very variable and determines the antigen receptor specificity of the molecule. And then down here at the C terminus in blue is the constant region of the molecule that doesn't change and is conserved. It stays the same for all different antigen receptors, except for some exceptions that we'll talk about in a few minutes. Now, the way B-cell antigen receptors, or immunoglobulin molecules, or when they're secreted antibody molecules, recognize antigen is illustrated on this slide. And what you can see is that the variable portion of the antibody molecule can recognize different shapes depending on the specificity of that receptor. So in this case, it's recognizing the little round red guy. In this case, it's recognizing a piece of the round green guy. In this case, it's recognizing a portion of the yellow antigen. And so these different shapes that the B-cell antigen receptor is recognizing are referred to as conformational epitopes. So conformational is just referring to the shapes of the molecular features. And that's how B-cell antigen receptors recognize antigen. So one of the puzzles about the way the system works is that I told you that there's millions and millions of different lymphocytes, each with different antigen receptors. And each of these individual lymphocytes has a single type of receptor with a unique specificity for antigen. But even though there's millions and millions of different antigen receptors, the estimated number of genes in the human genome is about 30,000. So if there's one gene for one protein, this kind of violates that rule. So how can there be millions of different antigen receptors when we only have 30,000 different genes in the human genome? And the answer to that puzzle is a process called somatic recombination. And what somatic recombination refers to is a process that occurs during both B and T lymphocyte development, where different mini-gene segments of a very large genetic locus rearranged together in different combinations that then will encode the antigen receptor specificity of the antigen receptor. So here what you can see is you have different mini-gene segments. In this case, in the dark red, they're abbreviated V gene segments, V for variable. And then here in the kind of salmony color are D gene segments, D for diversity, and J gene segments, J for joining. And what happens during lymphocyte development is that each individual lymphocyte, as it's developing, will pick one of these mini-gene segments in a more or less random way and splice out the intervening DNA. And so the part of the gene that encodes for the antigen receptor is made up of these different mini-gene segments that are all spliced together here. And this is the coding portion of the gene that encodes for the protein that in turn determines the antigen receptor specificity. And so this is a process that's unique to adaptive immune cells. So it's unique to T cell and B cell antigen receptor genes and only occurs during T and B cell development. Okay. The other thing I wanted to mention while we're talking about antibodies or B cell antigen receptors is that, as we'll see in later lectures, they're a very important part of the immune system in terms of providing protection from different infectious disease, and as also you'll see, can play a role in different types of autoimmune and inflammatory diseases or allergic diseases. Also, experimentally and clinically, antibody molecules are a key reagent for many assays that are used in laboratory testing, and also, as you'll see, as therapeutic reagents in the form of drugs. And so just to give you a couple of examples of the uses of antibodies in clinical laboratory testing. So one use of an antibody is in enzyme-linked immunosorbent assay type procedures that are used very widely in clinical laboratories for a variety of things. And this is just a cartoon to illustrate one way that you can do these types of assays, where you coat a plastic dish with antibodies, and then you add an unknown fluid. It could be serum that may contain different hormones. And if you coated the plastic dish with an antibody that recognizes, for example, human chorionic gonadotropin, if you want to do a pregnancy test, the level of HCG would bind to the antibody, and then you'd have a secondary antibody that you would add to the well that would in turn also bind to HCG in proportion to the amount of HCG that was present in the well. And so, and often attached to these antibodies, you may have an enzyme or a fluorescent molecule that will induce a color change. Again, and the degree of color change would be proportional to the amount of antigen that's bound in the well. And so, as I said, these are widely used tests that's used to quantify a variety of clinically important molecules that may be in serum or fluid or in other contexts. Another example where people use this type of technology is to use it to detect actually other antibodies. So if you want to determine whether or not somebody is potentially infected with the HIV virus, you can use antibodies that are specific for you can coat the well with an HIV-specific antigen, and then to see if somebody is infected and making an HIV-specific antibody, you can add their serum to that well, and those HIV-specific antibodies would bind to the plastic dish and cause a color reaction, and that would be indicative that the patient is infected with HIV. Another common example of where antigen-antibody interactions are used clinically is in blood typing. So as you may know, red blood cells come in different flavors, type A, type B, type AB, type O, and you can determine what type somebody is by using antibodies that recognize that specific blood group antigen. So in this case, you might add an antibody that's specific for red blood cell type A, and if this person's blood contained red blood cells with that type, the antibodies would engage with the antigen on the surface of the red blood cell and cause clumping or agglutination of the red blood cell. Whereas if you added antibodies that recognize the type B antigen, these antibodies would not interact with the red blood cell, and so no agglutination would occur. And so this is another very simple, very old test using antibodies to determine what blood type someone is. We also use monoclonal antibodies or antibodies as therapeutic reagents in a whole variety of clinical contexts. And this slide just illustrates a few examples, and there's many, many, many more examples, and more are approved by the FDA every year or every week, it seems like. And these antibodies may recognize specific cells and are used as a form of cancer immunotherapy. So if you had an antibody that was specific for breast cancer cells, as is the case with Herceptin, or antibodies that are specific for B-cell lymphoma cells, as is the case for rituximab, you give these antibodies to patients. They interact with the cancer cells and facilitate immune-mediated destruction of those cancer cells. People also use antibody targeting to target soluble molecules that T cells and B cells make to modify the immune response. So another common example is antibodies that interact with a cytokine called tumor necrosis factor that's used as one potential treatment for rheumatoid arthritis. And again, we'll talk more about this in later lectures. Okay. So now I just want to move on a little bit and talk a little bit about the structure and biology of the T-cell antigen receptor. So we've already talked about the B-cell antigen receptor, or the or antibody molecules, and the T-cell antigen receptor has a very similar structure. It's also a heterodimer made up of two different proteins that are attached to one another by interchain disulfide bonds. Just like the B-cell antigen receptor, the T-cell receptor has a variable portion that's involved in antigen recognition and a constant region that maintains the structural integrity of the molecule. But what's different about the T-cell antigen receptor compared to the B-cell antigen receptor is the form of antigen that they recognize. So remember that the B-cell antigen receptor recognizes shapes of antigens or conformational epitopes. What the T-cell receptor recognizes are small linear peptides that are typically derived from a much larger protein, and these peptides are presented to the T-cell receptor by another molecule called the MHC molecule, or the Major Histocompatibility Complex molecule. And these molecules are expressed on the surface of antigen-presenting cells. And so the MHC molecule associates with this peptide, which is in brown here, and what the T-cell receptor is doing is it's recognizing both the MHC molecule and the peptide presented by the MHC molecule at the same time. So there's multiple molecular interactions by the variable portion of the T-cell antigen receptor with both the MHC molecule and the peptide. And these MHC molecules are very important for regulating T-cell activation and the immune response in general. And so I just want to talk a little bit more about them and how they interact with these peptides and present them to T cells. And so there are two main classes of MHC molecules called MHC class 1 and MHC class 2. These MHC molecules in people are also known as the human leukocyte antigens. And as I said, these MHC molecules, because they present peptides to T cells, are critical regulators of the immune response. And so how do these peptides get presented by these MHC molecules and by these antigen-presenting cells? And so this is just a cartoon just to show you a very simplistic view of how this works. So this would be one antigen-presenting cell, and in this case, this antigen-presenting cell has within its cytoplasm a protein. And what happens is the protein gets degraded into peptides, and that degradation step is called antigen processing. And so the protein gets broken down and processed into peptides. And these individual peptides, or pieces of the protein, associate with the MHC molecule and are presented on the cell surface of the antigen-presenting cell. So this little red dot reflects the peptide, or the processed piece of the protein that's associated with the larger yellow MHC molecule and presented together as a complex on the surface of the antigen-presenting cell. And then the T cell and the T cell receptor would come down and recognize this MHC peptide complex. And so there are a variety of different types of antigen-presenting cells, but broadly we can classify those antigen-presenting cells as cells that are very good at antigen presentation, and we call these very good antigen-presenting cells professional antigen-presenting cells. And they're largely classified as three types of immune cells that we've talked a little bit about already: dendritic cells, macrophages, and B cells. And then other cells are less good at presenting antigen, and these cells are referred to as non-professional antigen-presenting cells. And these can mostly be thought of as any cell that has a nucleus that we have in our body. And all those cells express MHC class 1. And one of the things that makes professional antigen-presenting cells special is that they not only express MHC class 1, but they also express high levels of MHC class 2, and they also express special molecules that are referred to as co-stimulatory molecules that help promote T-cell activation. And we'll talk again more about the steps involved with T-cell activation in a later lecture. And so as I mentioned, there are these two main classes of MHC molecules, MHC class 1 and MHC class 2. And it turns out that these classes of MHC molecules process and present peptides derived from different sources. So MHC class 1 molecules present endogenously derived antigens. And so what that means is that they're proteins that are made and present within the cytoplasm of an antigen-presenting cell. And so an example of where this would be immunologically important is if a virus infects a cell and viral genes are transcribed and translated and new proteins are made, those new proteins, some of them, can be degraded into peptides and presented by MHC class 1 and presented on the cell surface. And so in this case, you'd have an MHC class 1 molecule presenting a virally derived peptide. And so this is important immunologically because it would allow a cytotoxic T lymphocyte that has a receptor specific for that viral peptide presented by this particular MHC molecule to recognize that cell and recognize that that cell is a cell that's infected by the virus. And so in this case, if this cell has cytotoxic effector function, it can cause lysis and killing of that virally infected cell. Another class of MHC molecules are MHC class 2 molecules. And remember, MHC class 2 molecules are expressed almost exclusively by professional antigen-presenting cells. And MHC class 2 molecules present peptides that come from proteins that came from outside of the cell. And so in this case, you have a cell that has phagocytic activity, and so it's internalizing an exogenous antigen. In this case, it looks like a bacteria molecule, and then it's degrading proteins associated with that bacteria into peptides, and some of those peptides are associated with the MHC molecule, and the MHC molecule is then expressed on the cell surface, where it can then engage with T cells that have receptors specific for peptides from that bacteria and facilitate activation of this cell that's phagocytosed the bacteria. And so MHC class II molecules present exogenously derived antigen that comes from outside of the cell. And so MHC class I and MHC class II presentation are associated with different immunological effector functions. And that's largely because MHC class I molecules interact with the CD8 protein that's expressed on CD8 T cells. And as I mentioned in the very first lecture, CD8 T cells are the type of T cell that tend to exhibit cytotoxic effector function. And so when you have a T cell receptor that recognizes MHC class I peptide complexes, this T cell will express the CD8 molecule, and that's good because the MHC class I molecule is likely to present peptides that came from virally infected cells. And so this will facilitate this effector CD8 T cell, its ability to kill this virally infected cell. Whereas in this case, you have another antigen-presenting cell that's presenting peptides presented by MHC class II, and MHC class II interact with the CD4 molecule. And so CD4, what that means is that CD4 T cells are especially good at interacting with peptides presented by MHC class II, and that allows the different classes of CD4 effector cells that, again, we'll learn about in the next lecture, to mediate different types of effector functions that are associated with MHC class II presented peptides. So again, just to reiterate this concept, what this means is that MHC class I presented peptides activate CD8 T cells, and that MHC class II presented peptides activate CD4 T cells. Okay, so let's talk a little bit more about the MHC complex and genetics associated with the different variants of the MHC gene that are inherited from our that we receive from our parents. And so one of the important things about MHC molecules is that they're very polymorphic, meaning that there's a lot of different variants of genes encoding for the MHC genes in the population. And these different genes are historically referred to as immune response genes. And historically, the MHC molecules were first identified as the major genetic locus that regulated the acceptance of tissue transplants. Because people realize that, at least in animal studies, and it turns out this is true in people as well, that you're more likely to be able to tolerate getting a transplant from somebody that has similar MHC molecules compared to getting a transplant from somebody that has very different MHC molecules. And the reason for that is if you have different MHC molecules, you recognize those MHC molecules as non-self or foreign and are more likely to mediate an immune response against those types of molecules. And so let me just talk a little bit about the variability of these MHC molecules. So these MHC molecules, even though they're fixed in the genome, meaning that they're inherited from your parents, they have enormous diversity. They're very polymorphic. So this is different than the diversity of B cell and T cell antigen receptors, which are diverse through this process of somatic recombination, or rearranging these different mini gene segments. So unlike antibodies or B cell receptors or T cell receptors, MHC molecules are fixed. You get what you get from the genes that you got from your parents. But MHC diversity is mediated through what's called genetic polymorphisms, meaning that there's a large number of different alleles in the population at each of the different MHC loci. And so this polymorphism results in a tremendous diversity of MHC molecules within a species. And so there's different types of MHC class I and different types of MHC class II genes. So this is just a bar graph illustrating the diversity of each of these different genes. And so just to start off with MHC class 1, because it's a little simpler nomenclature in people, MHC class 1, again, is what we refer to as HLA, and there's three main types of MHC class 1 in people: HLA type A, HLA type B, and HLA type C. And you can see for each one of these genes, there's a lot of different alleles in the population. And this is a slide that's several years old now, and so there's more alleles that have been identified over the last 10 years or so. But this is just to give you a rough idea that the different genes that encode for these different MHC class 1 proteins are very variable in the population. So there's approximately 559 HLA-B alleles or flavors of this protein that's in the human population. And the same thing exists for MHC class 2. In this case, it's referred to as HLA, and there's different HLA class 2 alleles or genes referred to as DR, DQ, or DP, and you can see that depending on each of these proteins, there's a different amount of variability associated with each of these proteins. And so there's a lot of variability associated with the HLA-DR protein and relatively less variability associated with the HLA-DQ protein. And so the importance of these different alleles or polymorphisms of the MHC complex affects the immune response in really a variety of ways. And so one way of visualizing this is just in terms of T-cell receptor recognition. And so these polymorphisms or different alleles change amino acids within the region of the MHC molecule that the T-cell receptor interacts with. So this is the MHC peptide binding groove, and imagine here's the peptide right in here. And you can see that the polymorphisms are clustered to affect the structure, the molecular complex that affects the specificity of the peptides that are within this MHC peptide binding groove. And not only that, it affects the contact areas where the T-cell receptor interacts not just with the peptide, but also with the MHC molecule itself. And this is true for both MHC class 1 and MHC class 2. And so there are a variety of examples where different alleles of MHC molecules are associated with different types of inflammatory diseases. And so, for example, if you have a human MHC HLA-B27 class 1 allele, you have an 87-fold greater chance or relative risk of developing an autoimmune disease called ankylosing spondylitis compared to the population that does not have that particular MHC class 1 allele. It doesn't mean that you're going to get ankylosing spondylitis, it's just that you have a much greater relative risk of developing that disease if you have HLA-B27. And so the important thing to realize when you're thinking about the disease association with MHC molecules is that the MHC molecule itself is not the cause of the disease, but there's often a strong association with having a particular MHC allele and the incidence of that disease. And this is just a table illustrating a variety of different autoimmune diseases where different MHC alleles, and this is true for both MHC class 1 as well as MHC class 2, confer change in your relative risk of getting these various different autoimmune diseases. The other area where different MHC alleles are clinically important and people actually test the MHC type of patients is in transplantation, especially in kidney transplantation and bone marrow transplantation. So this is a slide from large clinical studies looking at the chances of renal graft survival or how long a kidney transplant will last in a transplant recipient, depending on whether or not they're completely matched at MHC class 1 or MHC class 2, curve number 1, or if they have varying degrees of mismatches for MHC class 1 or class 2. And you can see that the two green curves, where they're more or less completely matched, you have a much greater likelihood of graft survival for a long period of time compared to kidney transplants where the MHC molecules, where there's a very significant mismatch, so curve number 6 or curve number 5. Again, indicating that the more similar you are at the MHC locus genetically, the more likely you would tolerate receiving a kidney transplant from an unrelated individual. So just to summarize then everything that we've talked about so far, In this mini lecture, we talked about really three main molecules that are critical in antigen recognition: the B-cell receptor, the T-cell receptor, and the major histocompatibility complex molecules. These molecules all belong to the same kind of large family of molecules called the immunoglobulin supergene family because they have a conserved structure. And especially for the B-cell receptor and the T-cell receptor, you have a variable portion that's involved with antigen binding and a more constant region down here. And all of these molecules are heterodimers, meaning that they're made up of complexes of two individual proteins for the most part. And for the B-cell receptor and the T-cell receptor, there are very many common features structurally that I just mentioned, but there's also some distinct features of the B-cell receptor and the T-cell antigen receptor in terms of the form of antigen that they recognize. So remember, B-cell receptors recognize conformational epitopes or shapes that may or may not be cellularly associated, whereas T-cell receptors typically only recognize small linear peptides that are presented by MHC class 1 or MHC class 2 molecules.